Greetings everyone, and welcome to another exciting genetic engineering and biotechnology news webinar. Our presentation today is entitled Establishing Fertility Biomarkers. Bill Galiskis, technical editor for GEN, and I'll be the moderator for today's webinar presentation. With almost 15% of couples at reproductive age needing reproductive technology assistance to conceive and 1/3 of female oncology patients at risk for developing premature ovarian failure, scientists are continually searching for improved methods to quickly and accurately assess a patient's current fertility status. Establishing valid biomarkers of fertility will provide researchers with advanced diagnostic insight into reproductive disorders, as well as normal biological functions, such as proper oocyte development, ultimately improving the outlook for struggling patients. In order to perform their work properly, fertility researchers are reliant on quality reagents, which begins with having a superior grade of the most universal solvent, water. Often taken for granted, consistent high-quality laboratory water can make the difference in improving sample preparation and obtaining reproducibles. Let's meet our panelists for this GEN webinar, who will discuss their research into identifying novel fertility biomarkers and how increased water purity has aided their experimental design and improved their laboratory workflows. Our first speaker today will be Maurizio Zuccotti, Professor of Developmental Biology at the Universities of Parma and Pavia in Italy. Dr. Zuccotti will discuss his recent findings about the biochemical pathways regulating the growth of mammalian oocytes and the cytological and molecular markers that could help identify the cells that are competent for sustaining development. Our second presentation will be given by Estelle Richet, Senior Scientist in the Lab Water Application Group for Millipore SAS in France. Dr. Richet will outline various water purification modalities and the potential impact water purification can have on laboratory research, such as Dr. Zuccotti's. Before our speaker, I want to encourage the audience to submit questions for our Q&A session at the end of the presentations. We'll try to get to as many questions as we can, so simply type your question into the Q&A box on the left-hand side of your screen and hit submit. All right, now that all the logistics are out of the way, let's begin our webinar and open up the floor to our first speaker. Maurizio?
Thank you, Jeff, for your kind introduction. Our search for fertility biomarkers spans through spermatogenesis and oogenesis and the early stages of pre-implantation development until the embryo implants on the uterine wall. Today, I will focus my seminar on female infertility. Because of the two genders, it is the one most affected by fertility problems. Just consider that every year, 1/3 of oncological female patients, which count for about 300,000 in countries like USA or 30,000 in Italy, France, or UK, may develop premature ovarian failure. And to these, we must add a 15–20% of couples which are infertile, and of these, 40% are attributed to female infertility. Clearly, the availability of robust methods of in vitro culture of ovarian follicles and of markers of oocyte quality would help the production of follicles and the identification of those oocytes that, after fertilization, are capable of further development, and thus improving the chance for women of having a baby. In our lab, we are working for trying to find markers of the oocyte developmental competence using the mouse as model animal. Finding markers of the oocyte developmental competence means understanding how the oocyte acquires during its growth within the ovary the capacity to sustain development. First, let me give you some background. Here you see a mouse ovulated oocyte surrounded by layers of companion cumulus cells which nourish the oocyte during its growth within the ovary. The idea that the egg supports the development of a new individual dates back to the 17th century when William Harvey wrote Omne vivum ex ovo, which we would translate in modern times as embryogenesis begins during oogenesis. This means that it is during oogenesis that the oocyte accumulates those goods, those factors that are needed to sustain the early stages of development. Importantly, whilst the ovary contains thousands of oocytes, only a handful of them will be ovulated, and only a very few of these will own developmental competency. Just as an example, in our species, of the 1,000 oocytes present in a woman ovary, only 1 per month will be ovulated during a reproductive window that lasts for about 40 years. Therefore, only less than 500 eggs will be selected for being ovulated, and of these, many will be developmentally incompetent. In other words, incapable of sustaining full-term development with the birth of a new individual. Our species has a high reproductive failure of about 65%, which means that only 35% of the fertilized eggs will give birth to a new individual. In short, studying the mammalian egg is not just a matter of improving our basic knowledge, but it has important medical implications. A major question we are addressing in our lab is what makes an egg good or bad. In other words, which is the cytological and molecular identity of the developmentally competent or incompetent oocyte. In the next slide, I'll show you our quest for markers of the oocyte developmental competence. To this end, we looked at different specific molecular and cytological aspects of the oocyte during its maturation phases, as listed in this slide. We studied the expression of specific transcripts of protein or the whole oocyte transcriptome to highlight transcriptional networks, or we looked at more specific cytological features such as the cytoplasmic movements observed at the light microscope. All these studies were conducted using a model study in which oocytes choose development at the 2-cell stage, a model that I'm briefly going to illustrate. In this slide, you observe the 8 main stages of follicle maturation as it occurs in the mouse ovary, from the tiny 10 micrometer in diameter primordial follicle to the fully grown antral follicle, the follicle that is ready to be ovulated. These latter follicles are those that we isolate by puncturing the ovarian surface with a sterile needle. The cumulus oocyte complexes are isolated and fully grown antral oocytes freed from the surrounding cumulus cells. When these antral oocytes are stained with the Hoechst supravital fluorochrome, we observe the presence of 2 distinct classes of oocytes that differ for their chromatin conformation. On the left-hand side, we observe an oocyte showing its nucleolus surrounded by a ring of Hoechst-positive heterochromatin, and for this reason named surrounded nucleolus oocyte or a senoocyte. And on the right-hand side, an oocyte with a nucleolus lacking this ring and thus named not surrounded nucleus oocyte, or NSN. Importantly, the two types of oocytes are present within the ovaries of all mammals studied so far, like rat, cow, monkey, pig, mouse, and human as well. Although the analysis of the chromatin organization is a gross parameter, it turned out to be very informative on the oocyte developmental competence. In fact, when fully grown antral oocytes are isolated from the ovary, stained with Hoechst classified, matured in vitro to the metaphase 2 stage, and inseminated, both types of oocytes are capable of fertilization and to initiate development. But whilst SN oocytes reach the blastocyst stage, and if transferred to a pseudo-pregnant female, they develop to term, and SN oocytes stop development at the 2-cell stage. On the left-hand side is a summary of the main steps of this procedure. Oocytes are isolated and stained with Hoechst in M2 medium, matured in alpha mem, inseminated in Whittingham medium, and embryos are cultured in M16. All the media used were prepared in-house in milk water. In summary, these experiments showed that those oocytes with an SN type of chromatin organization may develop to term, whereas those with an NSN type of chromatin arrest development at the 2-cell stage. We then wonder what does cause the 2-cell block in mouse NSN oocytes, whether there could be specific molecular markers, for example, specific transcripts, differential expressed in the two types of oocytes. And first thing, we looked for differentially expressed maternal effect factors. As I said before, during oogenesis, the oocyte stores all the goods that are needed to initiate development. The great majority of these factors are eliminated soon after fertilization has occurred, and only a small fraction of these, the so-called maternal effect factors, escape degradation. These maternal effect factors guide the transition from a maternal control of development to an embryonic control until embryonic genome activation occurs. Embryonic genome activation occurs at the 2-cell stage in the mouse, the 4 to 8-cell stage in human, at the 8-cell stage in rabbit, and 16-cell in sheep. If something goes wrong and embryonic genome activation doesn't occur, the embryos inevitably arrest development. To try to answer to the question, what does cause the 2-cell block in developmentally incompetent NSN oocytes, we first looked at the expression of maternal effect transcripts. Of a group of maternal effect transcripts analyzed, the only one that resulted differentially expressed was a gene named Stella. The slide shows the protocol used for the isolation and preparation of oocytes with a single-cell analysis of their transcripts using quantitative real-time RT-PCR. From top left to right, fully grown follicles are punctured from the ovarian surface and isolated in single drops of M2 medium, pipetted in and out using a handful micropipette whose size in diameter is about 100 micrometers, just a bit bigger than the oocyte itself. This allowed the careful removal of the surrounding cumulus cells until each single oocyte was completely devoid of cumulus cells, and it was isolated in a single drop of N2 medium containing Herx. Following classification under a fluorescence microscope, single SN or NSN oocytes were placed at the bottom of a PCR tube containing lysis buffer. This single-cell RT-PCR analysis showed a downregulation of Ostella expression in incompetent NSN compared to SN oocytes. You may read the details of this procedure in our published paper reported at the bottom of the slide. A differentially expressed Stella gene led us to study the presence of its protein. The right-hand side panel shows a representative immunoreaction using an antibody against Stella protein on antral SN oocytes. Notice the presence of the protein and an SN oocyte. Notice here the downregulation of the protein, and on their derived metaphase II oocytes. The absence of this maternal effect factor in NSN oocytes could explain their developmental incompetence. On the left-hand side panel, I summarized the in-house preparation of the slide required for single oocyte immunocytochemistry. First, a thick tape with a squared hole of about 3 millimeter side is placed and stuck at the center of the microscope slide. Then, a tiny drop of 5 microliter PBS is put within this cavity, thus creating a small pool in which the single oocyte is kept throughout the changes of media and reagents required by the procedure. Again, all the media were prepared using milky water, and a detailed description of the method is found in our published papers. Next, we reasoned that Stella is just one of a group of genes known for being directly regulated upstream by another transcription factor named OCT4. OCT4 is a master transcription factor, a marker of pluripotency. This slide summarizes some of its key aspects, the most important of which is that it is expressed in pluripotent cells and is downregulated upon differentiation. As for Stella, also the OCT4 protein is present in SN antral oocytes and in their derived metaphase II, whereas it is downregulated in developmental incompetent NSN oocytes and in their derived metaphase II. Being OCT4 a master factor that regulates many other genes, we looked for other candidates and wondered whether OCT4 downregulation in metaphase II derived from NSN oocytes modifies the expression of other OCT4 regulated genes. To this end, we compared by microarray analysis the gene expression in metaphase II matured from both types of oocytes. We used an Illumina microarray that compared about 39,000 gene sequences, of which only 380 were differentially expressed, 303 were upregulated, whereas 77 were downregulated in metaphase II derived from developmentally incompetent NSN oocytes. 25 of these 380 genes are known as OCT4-regulated genes, and 18 are assigned to apoptosis or mitochondrial dysfunction. Overall, these results indicate the existence of an OCT4 transcription network whose regulation is critical for the oocyte developmental competency. In our work to come, we plan to find out when, during folliculogenesis, this transcription network is built up and how it evolves during preimplantation development. Technically, this is quite a tricky thing to do. It's a challenge since we will have to inject within the ooplasm of the growing follicle a construct that inhibits the OCT4 transcript transduction without harming the oocyte. And then the follicle will be grown up to full maturation and fertilized to observe further embryonic development. This is an experiment we have just begun, and we will keep you updated on this. All the transcript and protein markers described so far, whether it is STELA, OCT4, or those OCT4-regulated genes, give information on the molecular players involved in building up the oocyte developmental competence. However, they are invasive markers as they require for analysis the fixation or even the destruction of the oocyte itself. Therefore, although they are helpful in the identification of biological differences between competent and incompetent oocytes, they are, at the moment, unsuitable to be used in the clinic whenever one needs to identify those competent oocytes for improving the outcome of the use of artificial or reproductive technologies. The slides that follow will focus on two attempts we carried out to identify non-invasive markers that may distinguish the two types of oocytes. I'll first describe our work on the cumulus cells surrounding the oocyte, and then I'll show you our work, our microscope observation of the oocyte cytoplasm movements occurring during maturation. Cumulus cells surround the oocyte even after its ovulation has occurred and are dispensable for fertilization. In other words, they can be picked and analyzed separately without damaging the oocyte. And this is exactly what we did. As I have shown you in a slide earlier, fully grown antral follicles are isolated by puncturing the ovarian surface, and then, using a sterile micropipette, the two somatic and germ cell components are separated and placed in different single drops of medium. Next, the oocytes are stained with Hoechst and classified based on their chromatin organization into SN or NSN. The cumulus cells of about 40 SN oocytes or 40 NSN oocytes are then transferred separately into PCR tubes for retrotranscription and microarray analysis. And these are the results we obtained. The comparison between NSN cumulus cells versus SN cumulus cells highlighted the presence of 422 differentially expressed genes. 412 were downregulated and 10 upregulated. Of these, the anti-Mullerian hormone gene and HAS2 were the most up and down regulated respectively in the cumulus cells of developmentally incompetent NSN oocytes, and thus the transcripts of these genes are good candidates to become non-invasive markers. We are now working on trying to set up an RT-PCR assay capable of identifying their expression in the cumulus cells of single oocytes. Next, we studied the changes to the cytoplasm movements occurring during the maturation of the antral oocyte to reach the metaphase 2 stage. As I said earlier, this process occurs in vivo at the time of ovulation, and it can be obtained in vitro during about 9 hours of culture. Cytoplasmic movements are recorded using time-lapse imaging inside a biostation, which has a small incubator that keeps the temperature at 37 degrees centigrade, and the atmosphere with 5% CO2, which is the ideal environment for cell growth. The time-lapse recording obtained is analyzed with the cell particle image velocimetry method, which is a mathematical tool that allows a quantitative measure of the velocity of the cytoplasm movement. Following is an example of a movie that shows the measure obtained observing a developmentally competent SN oocyte. The two main movement peaks occur right at the beginning of oocyte maturation, at the time of germinal vesicle breakdown and at the time of the extrusion of the first polar body. The latter is a sign of meiosis resumption. When compared to SN oocytes, what strikes is the late extrusion of the first polar body. Overall, the analysis of 127 SN and 109 NSN oocytes brought up differences in the cytoplasmic movement profile of the two types of oocytes, particularly during 4 main time-lapse periods. Whilst these differences are evident when analyzing large numbers of oocytes, they cannot be used to identify single oocytes. But for this, we were helped by our bioengineer colleagues who developed an artificial neural network that, based on the results obtained with the particle image velocimetry, is capable of making a prediction of each single oocyte, classifying it as SN or NSN based on its cytoplasmic movement. In summary, the following are the main take-home messages I'd like to deliver with my webinar. First, within the antral compartment of the ovary, there are oocytes with a distinguished chromatin organization and are named SN or NSN oocytes. Those that have an NSN chromatin configuration are developmentally incompetent, whereas those with an SN chromatin may develop to term. Also, developmentally competent SN oocytes display a high expression of the stellar maternal effect factor, whereas in NSN oocytes, stellar is downregulated. The same profile of expression occurs for the OCT4 pluripotency marker. The downregulation of OCT4 correlates with the upregulation of a network of apoptotic genes which could mark the developmental incompetence of the NSN oocyte. The analysis of gene expression in the cumulus cells surrounding the oocyte could be a good non-invasive marker to distinguish between competent and incompetent oocytes. And finally, the analysis of the cytoplasmic movements occurring during the maturation of antral oocytes to metaphase 2 is a non-invasive marker of oocyte developmental competence. But to me, the most important take-home message of our work and experience is that the future of embryology and of biology as a whole lies in the collaborative work of biologists, biotechnologists, bioengineers, physicists, chemists, and physicians towards a common goal that in our lab we are trying to reach as part of the Center for Health Technologies, a center that groups labs that work on these different disciplines. Before I finish, I'd like to thank the people, past and present, of the lab, Professor Silvia Garagna, who's heading our group, and our three main collaborators, the bioinformatician and engineers, Professor Riccardo Bellazzi and Dr. Lorenzo Fassina of the University of Pavia, and Professor James Adjaya of the University of Düsseldorf in Germany. Thank you very much for your attention.
Thanks, Maurizio. That was a great presentation, and you provided the audience with a very nice snapshot of your research into uncovering biomarkers for oocyte development. Before we move on to Estelle's presentation, I want to remind everyone once again to submit questions for the Q&A session at the end of the next presentation. Simply type your question into the Q&A box on the left-hand side of your screen and hit submit. Both of our speakers will be available to answer your questions after the final presentation. So let's get our next presentation underway. Estelle, the floor is yours.
Yes. So for the next few minutes, I'm going to talk to you about water. I know this is maybe something that's a bit unusual for you because maybe you don't think that water is so important, but it actually is, because water is probably the solvent you use the most in your lab. Maybe sometimes without even thinking too much about it. So in the next few minutes, I will try to convince you that actually water quality is really important for your experiments and that it's selected very carefully. All right, so if you think about what you use water for in your laboratory, probably the most common use of water is the preparation of buffers and reagents. Or if you dilute buffers that you purchase that are concentrated and things like this. Another very common use of water is to prepare cell culture media. Or maybe if you don't prepare your own media, you might be using water to prepare the additives that you add to your media. So if you think about it, in all those cases, water is actually probably 90-some% of your final solution. So of course, if you were to have some contaminant in your water, it may have an impact on your final experimental results. So that's something to really watch out for. Okay, so other uses of water might be preparing your samples, diluting them, dissolving them, for blanks, for preparing standards. And another use of water that many people don't think much about either is washing and rinsing containers and glassware in the lab. And here again, maybe this is not even done in your lab. Maybe this is done somewhere else, downstairs or in a washroom. But are you really sure that your glassware is as clean as you think? And really, if water of lesser quality is used to rinse your glassware, maybe it will leave a few traces of contaminants that will then carry on into your solution and may have an effect on your final results. So really, it's very important to select the right water quality for everything you do in the lab, even including washing and rinsing glassware. So there are many different types of laboratory water available. I'm sure in your lab you have several kinds of water. Maybe you have bottled water, maybe you have a distillator, or maybe you have a tap, next to your regular tap water that delivers deionized water, or maybe you don't even know what kind of water it is because it's available in your building, but you don't exactly know how it's purified. So the question is, how to select the best type of water for what you are doing in the lab. And is there just one best type of water, or do you need to adapt the water you use to what you're doing in the lab? About which one is best for doing what. All right, so if you think about your coming out of your tap, of course, it's mostly H2O, right? It's mostly water. What else is in there? Well, we group them into different families. So the first family I would like to talk about is bacteria. Of course, in your tap water, there are no dangerous bacteria, right? Because it is treated so that it's absolutely safe to drink. However, there are bacteria everywhere in our environment. Therefore, there may be a few bacteria at the tip of your tap that are harmless to your health. You can drink that water absolutely without problem. However, if you use that water for experiments, it might cause a few problems. Another family of contaminants that may be present in your tap water are organic molecules. So those molecules, they come from nature, from the plants and the leaves that may be falling into the water that is used to generate your tap water. There may also be some small amounts of residues of, pesticides maybe, or even medications. Nowadays there is more and more talk about the fact that there are a few medications that lurk in very small amounts in our tap water nowadays. So of course, this is all regulated, the levels are very, very low, so it's not bad for your health if you drink it, but once again, it may be harmful to your experiments, which may be much more sensitive to organics than you are. A third family of contaminants is particles. So there may be a few pieces of dust and sand and things like this in your tap water. But if you're doing experiments where you need to visualize, those might be a problem. A fourth family of contaminants are ions. So those also come from nature, from the fact that water comes into contact with many different rocks and the soil. And once again, those are actually quite good for our health, but they may have an impact on your experiments. And the third family of contaminants are gases, dissolved gases. So all those small amounts of contaminants that are present in your tap water are not dangerous for your health. However, they're not so good for your experiments. And that's the reason why usually you will not even think about using tap water for experiments, and you will use water that's a bit more purified. Okay, so there are different types of purified water available. So in order to make things a little bit easier, usually manufacturers of water purification systems divide the waters into 3 different types. So the type 3 at the bottom of this pyramid is water that is really greatly purified. In most cases, it's already purified up to 90%. This is, for example, the type of water that is generated by reverse osmosis. So we call this pure water, and the quality may differ depending on the water that you start with. And this really this type of water quality may be used for really basic needs in the lab, some rinsing of containers that are not so critical, in heating baths and things like this. Now, a type of water that's a bit more pure is type 2 water. And that water is a quality that's much more reliable. So usually it's obtained by a combination of different water technologies, such as, for example, a combination between reverse osmosis and electrodeionization. And this water can be used for many different applications in the lab, like preparing some buffers, preparing media for microbiology, preparing solutions for histology staining, and so on. Finally, the best type of water, the most pure type of water, is type 1. We also call it ultra-pure water. So that one is really contains only H2O, nothing else. And this is what we would recommend to use for really very sensitive, very delicate types of experiments. So, for example, for cell culture, for molecular biology, or for all sorts of analytical techniques like LC-MS or ICP-MS, for example. All right, so how is water purified? Well, there is not one single purification technique out there that allows you to have directly extremely pure water from tap water. So in many cases, the way water is purified is in 2 steps. The first step is called pretreatment, and this really removes the biggest amount of contaminants, over 90% in most cases. And then the second step is called the polishing, and that step really removes the traces of contaminants that are left in your water in order to generate ultra-pure water. Some systems nowadays actually combine those 2 steps into one purification system, which is really very convenient. So to give you an idea, most water purification systems have a way to monitor the levels of contamination that's in water. And in most cases, they can monitor both the amount of ions in the water and the amount of organics. So if you consider that in tap water, you start with ppm, or parts per million, of ions and organics. Then after the pretreatment step, you're down to below the ppb level, so below the micrograms per liter level of ions, and about below 50 ppb of organics. And then if you look at ultra pure water, extremely pure water, then you're down to parts per trillion or below of ions, and usually below 5 parts per billion or micrograms per liter of organics. So depending on what you need to do in the lab, you can pick different types of water. It is possible also to add additional purification after this system by just adding a little cartridge at the end of the point of delivery. So in our case, in Dr. Zuccotti's laboratory, he has 2 different types of filters. On one of his point of delivery, he has a screen filter called the Millipack, and this one is just a 0.22 micron filter which will reject particles and bacteria. On another of his point of delivery, he has an ultrafilter, which is a nice technology that removes endotoxins, nucleases, and bacteria. And of course, this is really recommended for example, molecular biology or cell culture applications. So he has the possibility to take one type of water or another depending on what it will be used for. All right, so why is water purity important, and what can you do to your experiments? All right, so one thing that's very important in Dr. Zuccotti's lab is cell culture. So if you are preparing cell culture media, I know you know that cells are very sensitive to their environment, whether it's the media itself or the temperature, the atmosphere, and so on. And most of us are mostly concerned about the biological contamination of media. Like, if there are yeast or bacteria in the media, it can really have drastic effects on the cell culture. So that's the reason why we usually autoclave media. But there is another type of contamination to take into consideration that is not as visible because, it doesn't grow, it's not visible under the microscope. It's chemical contamination. So for example, if you use water to prepare your media that contains a small amount of metals that are toxic to cells, for example, or a small amount of pesticides, that come from the tap water, or even endotoxins, then it may have an effect on your cells. And the problem is, it may not kill them, it may just affect slightly their metabolism, and you may not realize it right away. And you may only realize it later on and have to troubleshoot and maybe rerun some experiments. So this is the reason why it's really important to use high-quality water to prepare media. And in the case of Dr. Zuccotti's lab, it is even more important because he is working mostly with embryonic cells, gametes, and these cells are extremely sensitive, even more sensitive than more mature cells, to their environment. Of course, to do the work that he does, it's really important to have extremely skilled scientists, but also it's very important to use really high quality and high purity reagents, including the water. So for preparing his media, he uses molecule water that has been further purified by an ultrafiltration cartridge, and he obtains really very good quality results. Another type of experiment that is performed in Dr. Zuccotti's lab is immunocytochemistry. So here also water quality may have a big impact on results. It's just that maybe it's slightly different types of contaminants that are important. For example, of course, anything that could deposit on the slides and create artifacts would be really a problem. So particulates and bacteria should absolutely be avoided in the water that is used to prepare all the solutions and reagents that are used for immunocytochemistry. Also, organic molecules, some of them may have an effect on the intensity of fluorescence, so this is something to keep in mind. And chlorine may have an effect as well, and it may actually have a bleaching effect, so you may not be able to see what you expect to see or not have the same intensity. Finally, some inorganic ions may have an effect on the pH and also affect the development of the fluorescence. So really here as well, using ultra-pure water is really important in order to get optimal results. One last technique I wanted to discuss briefly that is used also in Dr. Zuccotti's lab is quantitative PCR. And here, of course, the most important contaminant to take into account is nucleases, as well as ions such as magnesium, of course, which is always very important for PCR. And some metals as well may interfere with the polymerase activity. Some organics may also interfere with the DNA or with RNA. So really here as well, high purity water is very important, but here it's important to use high purity water that does not contain nucleases. And one good way to obtain this is to use the ultrafiltration final filter, and this is just really much more convenient than doing a DEPC treatment, which takes time and is a bit cumbersome. So really with one system, he can have water that can be used for many different types of experiments in the laboratory. So as a conclusion now, I just wanted to recap a little bit. So I hope that I convinced you in these few minutes that you should really look at the quality of the water that you're using in the laboratory, and that water quality is really important and that it needs to be adapted to the type of experiment you're doing. And you should look at it like all your other reagents in the lab, and really make sure to use high quality water. Another thing is to adapt the water quality to the type of experiment you're doing, and take into consideration the specific contaminants that may have an impact on the type of experiment you're doing. And finally, it can be very convenient to have a water purification system in your lab, because you can actually tailor the water you will obtain from it to the type of experiment you will do by adapting different types of final purification to your point of delivery. In addition, I would like to remind you of something that's really quite important, which is that it's important to really take good care of your water purification system and maintain it well in order to get the best results at the end and to have the highest quality. And it may also be a good idea to have some preventative maintenance, for example. And there are many companies out there that have highly skilled engineers that can help you to do this. And finally, I wanted to mention that it is now possible to have software that can track your water quality over time. If this is something that you feel would be helpful to you, this is now possible. So this is all from me. Thank you very much for listening. I will now open the floor for questions. And if you would like to ask me additional questions, don't hesitate to email me at this email address. And I would also invite you to have a look at our website, where you can find a lot of very useful information. Thank you very much.
That was excellent. Thanks very much, Estelle. I think the audience is now extremely cognizant of the impact water quality has on improving laboratory results and reproducibility. Before we start the Q&A session, I want to let everyone know this is your final chance to submit questions for our speakers. We already had some really great questions that have rolled in, but I'm encouraging all of you in the audience to keep them coming. All right, so while everyone is submitting their final round of questions, if you could bear with us for a few moments while we get all the questions together and get everything situated. All right, everyone. So just before we begin the Q&A, I just want to point everyone's attention to the resource list box on the bottom right-hand side of the screen. Something a little write-up by one of our speakers, Estelle, as well as some of our colleagues. And so if you wanted some more information on some endotoxin-free water for tissue culture, you can click there, and there's a nice PDF for you there. So with that, let's get to the first question. The first question is for Maurizio. Maurizio, what is the frequency of SN and NSN oocytes in the mouse ovary?
In the mouse ovary of a young female, it is about 50% for both. But these frequencies change during aging, i.e. The percentage of SN oocytes increases and the percentage of MSN oocytes decreases.
All right, thank you, Maurizio. We have another question for you. One of our audience members would like to know, what about using the fluorochrome host instead of a biomarker that distinguishes between developmentally competent and incompetent oocytes?
The host is a very good biomarker, but it can't really be used outside animal models, because it's a fluorochrome that goes into the DNA, and it can be mutagenic. Therefore is not suggested for its use is not suggested for human purposes. And this is one of the reasons why we are looking for other types of markers, and non-invasive markers particularly.
All righty, thank you, Maurizio. Estelle, we have a question for you. Our audience member would like to know about the special filter that removes nucleases. They would like to know how does it work and how long does it last?
So it's called an ultrafilter. So basically what it is some very fine membranes that are inside a cartridge, and the filtration is done simply by size. So really large molecules cannot go through, and very small ones can go through. And that way you can retain large molecules like endotoxins, nucleases, those large enzymes. So it's very easy to use. You just screw it on at the outlet of the system, and it can work for 90 days, and then at the end you can just discard it and use a new one. But it's very nice. You can replace the EPC treatment quite well, actually.
Great, thank you, Estelle. Maurizio, we have another question for you. Audience member asked, do you know how early in folliculogenesis OCT4 and Stella are expressed? Can we predict oocyte quality before germinal vesicle formation using these markers?
Mm-hmm. Yes, we do. We isolated oocytes throughout folliculogenesis and found that the very tiny ones, those that have a size in diameter of about 10-20 micrometer, do express the OCT4. But when oocytes reach the size of about 40-50 micrometer, OCT4 is expressed only in SN-oocytes and is downregulated in NSN-oocytes. Stella is unexpressed until the oocyte reaches the size of 40-50 micrometer, and then it's found only in, again, as for OCT4, only in SN-oocytes. Therefore, the answer to the question is yes, we can distinguish between the two types of oocytes earlier in folliculogenesis, and we actually published this work. And if the people that have asked this question would like to have the reprints, they can just write to me an email.
Thank you, Maurizio. And stay with us. We have another question for you. Is it possible to do Western blot analysis on a single human or mouse oocyte, or is it necessary to pool all of them?
Right. Well, thanks. This is a very important, very nice question, and it's actually what we are trying to do in our lab. And there is a very important paper that has just come out, and I can give the attendees the paper where it was published. It's called Single Cell Western Blotting, and it was published in Nature Methods, volume 11, page 749. And it's a very nice paper that describes the possibility of obtaining single-cell Western blotting, which can actually, with very difficulty, be done right now. And it's what we are trying to set up in our lab. It's a very important issue, this one. Thanks for the question again.
Right. Thank you, Maurizio. And we have one last question for you and for their webinar. About the collection of mouse oocytes. Did you select them at the stage of estrus? Right.
I didn't mention this. The oocytes were selected 48 hours following injection of an hormone called PMSG, which is a follicle-stimulating hormone. So, because that's the best timing where you can collect oocytes for maturation in vitro and further fertilization and embryonic development.
Alrighty, thank you, Maurizio. And with that,
Sure.
we've come to the end of our webinar. So I'd like to remind everyone that the webinar will be archived for up to a year on our website at www.genengnews.com. So if you missed any parts of it, you can watch it again, or feel free to forward the link to your friends and colleagues, which we always recommend. I'd like to thank Maurizio and Estelle again for their informative presentations. Excuse me, and I'd like to thank the audience for their attention and thoughtful questions. And a very special thanks to Millipore Sigma for sponsoring this webinar. So hopefully we'll see you again at another Gen webinar in the near future. Now,